I was pumping my friend this morning for information about David. So David's a professor. He's from Ohio, with his bachelor's degree at University of Cincinnati. Master's and PhD from Yale University. And fun fact, 2 fun facts. He has over 5,300 citations of his work. Not sure if you knew that, but Google tells you a lot of things when you Google folks. Second fun fact is David has been involved in a coup and overthrown the president of the University of City College and has been the president of the Senate of Faculty for several years. So has a life outside of, you're looking at me, Miles, like, what is going on here? Has a life outside of the lab that involves some revolution. So you've been hearing about generative AI, you've been hearing about ChatGPT. We're going to get into a little bit of that when it comes to life sciences. So without further ado, David.
Thank you. It was a bloodless coup, by the way, in case you were wondering. All right. So it's a great honor to be here and to talk to you about our work a little bit. And I'm going to start off by just saying a little bit about generative AI. Generative AI is far too big of a topic to discuss in 30 minutes, so no justice will be done today. But thank you for coming along and for being here. So as I understood it, most of you guys are working in the life sciences. Some are not, though. It's okay if you're not. So I'm going to try to address everybody today, if you're in the life sciences or you're not. So what I was asked to talk about is generative AI and how it is used in the life sciences. And the big way that we use it is a computer program called AlphaFold. And AlphaFold is a revolutionary calculation that allows the three-dimensional structure of a protein molecule to be calculated. And this is a long-sought goal and achievement of biologists for more than a century, to understand the structure of a protein and then to be able to just type the protein sequence into a computer and out comes a structure that is more or less correct. Probably not the full story, but more or less correct. This is something we've never had before, and I'll show you how we're using it in our work. Now, so let's start by just understanding what generative artificial intelligence or generative AI is. And so what I did is I went to ChatGPT and I asked ChatGPT to tell me, what is generative artificial intelligence? Back on track. Generative AI, what is it? So generative AI uses algorithms and models to generate new and original data based on patterns that it's learned from data that's been given to it. So these models are used to understand and capture the underlying structure and distribution of training data. So it's important to have enough training data. And then, importantly, this generative AI can generate new data samples that are similar to the training data, but not identical to it. And then this involves learning, capturing dependencies, and so forth. Now, generative AI has a wide range of applications, as you probably know, and we'll talk about one today, but some of them are listed here: image synthesis, video generation, data augmentation, and so forth. Now, it's not an exaggeration to say that this is the transformative technology of the 21st century. And it will have many positive aspects, and like any technology, it can also be misused, and it's already been misused a little bit. And it'll be up to you to distinguish between the two, and I would suggest staying on the side of using it in ways that benefit society. So you've probably heard about ChatGPT, of course, but there are many other applications of generative AI. Now, of course, ChatGPT is able to beat all of us at the SAT, LSAT, GRE. Seems to have a problem with English literature, so it's still not there yet. And then accounting, apparently. Not such a good accountant. Medical diagnoses, MBAs, it will write an essay for you, and it's a pretty good essay. And then this one, I like this one, that will be a sommelier for you and pick the wine for you. So this is really, it's the same, the underlying technology is to take a bunch of data, and the algorithms process the data, look at the patterns, and then use those patterns to make predictions. And it's getting better and better and better. So now, one of the groups that's paid a lot of attention to the development of ChatGPT is a company called Chegg. And you guys know Chegg, right? Chegg shares dropped more than 40% after the company says ChatGPT is taking its business. Chegg is a place where you can go to get exams and get some help, and now you can do it somewhere else. Now, the technology is has potential to do harm to humans, and so this has been highlighted recently by several prominent businessmen. Yuval Harari is a historian, and then the general idea of artificial intelligence in society has been the subject of a hearing before the Subcommittee on Privacy, Technology, and the Law. And so this is very much something that society is concerned about, particularly the negative uses of artificial intelligence. And if you saw the hearing about 10 days ago, you will know that before the hearing, a recording was played of the chairman of the committee, the voice of the chairman of the committee saying things that the chairman never said. And this was just to illustrate the power of artificial intelligence to produce sounds and voices that sound exactly like us, but aren't us. And of course, you can imagine the nefarious purposes that this could be used for. So this is something to understand. Now, several prominent voices in the field have called for a pause on the technology, and Jeffrey Hinton, who is, the New York Times referred to him as the godfather of artificial intelligence. He developed some of the early technologies for image analysis and for making images, and working with Google. He is now quit, and he's adding himself to voices calling for a pause on the development of artificial intelligence. And we're going to have to see how this works. The genie is out of the bottle, so to speak, and it will be up to our leaders and ourselves to consider how we're going to use this technology in a way that benefits society. Now, you might think that the artificial intelligence is new. It's actually not new. It dates back, the idea of artificial intelligence dates back to the 1950s, long before any of us were born. And Alan Turing was a computer scientist who thought about these things, and he developed a Turing test, which is it's a test to see if one could tell whether one's speaking to a computer or not. And so what I've listed here are examples of artificial intelligence that you probably already use. So email spam detection uses artificial intelligence. Google, you use Google Mail. And if we didn't have this, you'd have a lot more mail. So an artificial intelligence-based program was the first to beat a human at chess in 1997. And then the game Go, which is a considerably more difficult game, took longer but now a computer can beat a human playing Go. But look at all the rest of the things. So chatbots, fraud prevention, shopping recommendations. If you're on Netflix, you get the recommendations. Those are all based on your preferences. Agriculture, where to plant things, navigation, GPS. We're going to see them in self-driving cars. What I've been using are some writing tools that are interesting to improve writing. We have a lot of meetings, and then we want to have a little summary of the meeting. So voice to text has gotten a lot better, and now with artificial intelligence, it'll summarize the content of the meeting with just some bullet points. Medicine, of course. A colleague, a friend really, who I went to high school with, started a company that will make narratives out of data. So if you give it the baseball scores, the baseball line, it'll produce a story that can be published that is human readable, that describes the game. So that's narrative from data. It's another example of AI. Now, of course, ChatGPT is in the news. There's also DALL-E, which will make you an image based on words that you provided. I'm not going to show you any of the images that I made. Some of the ones that I've been playing with, and I think there's more improvements to be had, are a site called Tome.app. Tome.app will make you a presentation, and I asked it to make a presentation for you today. I had to go back to the drawing board. Science Summary will take a scientific paper and write you a summary, a brief summary, and it's pretty good. I cannot recommend Clear Brief. Clear Brief is will take opposing counsel's brief. Let's say you're involved in a court case. It will take the opposing counsel's brief and highlight weaknesses in the arguments. So the site says it will find whether there's hearsay or other kinds of errors that the opposing counsel has made. And you guys could probably do that as well. And then we're going to talk about AlphaFold. Now, I just want to emphasize something that AlphaFold and all these AI applications are really just about technology. And as a human society, we have lots of technologies. Everything that we've invented to help ourselves do whatever it is that we want to do, each one of them has a beneficial purpose, and each one has a nefarious purpose. So the question is, what's the impact? But there should always be an ethical component to use of technology, whatever the technology is. And that's something that each of us has to decide for ourselves and for society. So with that as a bit of an intro to AI, let's now jump into our work into biology. So structural molecular biology or molecular biology has its genesis in the period after World War II. And so many scientists who worked on scientific projects during the war, when the war was over, they turned their attention to biology, particularly physicists. And the idea was to try to change biology from something that's more descriptive to an understanding of biological function in terms of the molecules and atoms that carry it out: carbon, nitrogen, oxygen, phosphorus, sulfur. And a major focus of that effort was to try to understand the protein. Protein is a complicated molecule that carries out biological function. And so beginning in the 1950s, efforts were made to understand protein structure. And so proteins, as you may know, should know, are linear chains of amino acids. And amino acids are just depicted here as these little colored circles, but the gray line links them all together. And the different proteins differ from each other by having a different sequence of amino acids. And then the idea is that they fold up into a compact structure. And that compact structure is dictated largely by the sequence. So just without getting into a lot of details, the amino acids can have what's called polar. Polar side chains are happy in an aqueous solution in water, and we're mostly water. And non-polar are less stable in water. And so what you see in the folded protein is the green things end up in the middle, away from water, and the colored things, the red and blue, are on the outside. So this is something that we've observed in folding a protein, but this is really not enough for us to predict this structure. And what we'd like to do is be able to write out a sequence of these colored circles and get this. And this is the protein folding problem. So we're not able to do that for many years. And so instead, what were developed were several approaches to see proteins. Now, we can't see proteins with our eyes, but the way that you see a protein is to do something that I call atomic photography. And I saw when I was in my meeting today, there was a picture of a magnet. And I didn't hear anything, but the magnet is one way to see a protein. The way that my research group sees proteins is we use something called X-ray crystallography or electron microscopy to see proteins. Now, there's not enough time today to tell you what those are, but that's how we see proteins. Now, we can't see proteins the way that I can see you, because our visual system has a visual acuity of about 100 microns, and most of us don't even do that well. We can use a fancy tool here like a microscope to do a little better. The best ones can see down to about half a micron, but proteins are smaller than that. Atoms and molecules are 0.1 nanometers and 10 nanometers, so 1,000 times smaller than what we can do with light, the light microscope. And hopefully this is something that you all know. This is 10 orders of magnitude in which biology takes place in. So we live in a meter world. You probably have met some of these guys here, maybe. Millimeter, they're millimeters, like roaches or ants or other crawling things. We had an unwanted guest a few years ago. That's in the micron world. It's a little bigger than micron. Microbes, this is E. coli here. This is about a micron. So a micron is 1 million times smaller than us. Now, DNA is 1 million times smaller than a bacteria. It's 1 billion times smaller than us. And atoms and molecules are smaller. So light microscopy, because of the physics of light, allows us to more or less see down to the micron world. And it's X-ray, NMR, and electron microscopy that allows us to visualize this. Now with these techniques, we don't see the way we do with our eyes. The image just doesn't appear. We have to do a lot of work to get these pictures. But eventually we get a three-dimensional picture. Now, over 55 years or so, scientists have produced 200,005 protein structures. This is the protein data bank, and the protein data bank was started in 1971. And since 1971 to this day, we've produced 200,000 structures. That seems like a lot, right? That's 50 years. So where does that stack up with the available number of proteins? So here you see that you saw amino acids. Proteins are linear chains of amino acids. A protein can have hundreds of amino acids. And we know that humans have 200,000 proteins in them. Sequence the genome. But on planet Earth, there are 200 million distinct proteins, which means we've figured out the structure of 0.2% in 50 years. That's not so good. So this is painstaking experimentally determining these structures using X-ray, NMR, or electron microscopy. And each one takes from months to years to do. So ideally, what one would have is to have a computer program that you could just type the sequence in of the molecule, and out would come the structure. And this is what we now have with AlphaFold. And AlphaFold is able to take every protein that is known to science, which we are about 200 million, and spit out a structure. Now, many of these proteins change shape during their function, so the algorithm is not going to be able to do that, but at least it gives you a starting point to begin to understand motion. Now, how do we use AlphaFold in our work? So, one of the problems with the experimental techniques, NMR, X-ray, and cryo-EM, is something we call resolution. So resolution is how good is the image that we produce. And we always want to have the best resolution possible. We want to have the best and sharpest image, but we often don't get it. So an example is shown here. So we all have camera phones, and you take one of the older phones that doesn't have a great camera and take a picture, and you expand the picture, you can see the pixels. So this is what we refer to as a low resolution image. And you can tell that there's something here. There's a dense object over here, and there's some less dense and more dense, but there's no inferences or analysis possible with this image. So often, the images that we produce of our molecules look like this. This is an analogy. They look like this, and we can kind of say, well, it's this wide and this tall and so forth, but we don't really understand how it works because we're limited by the resolution. So now I'm going to get a better camera, get more data, and now it's the same picture, but it's got more information in it, more resolution, and now you can see that there's something here, and the dense object has gotten a little clearer. This looks like a window, and it looks like perhaps there's a building behind here. And so, but still, it's not very satisfying, and you'd like to see kind of a very sharp image of this. So then I get one of these very fancy cameras that gives me a very sharp image, and now I can see the object in all of its detail. Windows, it's a statue. I can read what's written here, which I couldn't do before. So now these details allow me to begin to think about this object. What's it for? What's it doing? Why is it here? So the same process is, we use the same process in thinking about a molecule. Now, the molecule that we've been studying recently is involved in the replication of DNA. What you're seeing here is a cartoon representation of the machine that replicates DNA. So if I start this, turn this machine on, what you see is things moving around, but the thing to pay attention to is that the input parent duplex comes in at the top. The two strands are separated from each other, and they provide substrates for the yellow objects, which are polymerases, and the polymerases copy one strand into the next strand to make a second duplex. So one duplex comes in, two come out. Every cell that divides is doing this. Now, the thing that I've studied different parts of this machine over my career. The thing that we're studying recently is this thing right here, which is this magenta. It's in the background minding its own business, but it does a very important job. And what you can see is it's taking one of the strands within, it's a ring-shaped protein. It's admitted one of the strands into the ring, and it's excluded the other strand. And this protein is called a helicase. And what it does is it helps to separate the two strands from each other, which would otherwise be stably attached to each other. So this is a motor. It's traveling along one of the strands. As it's doing that, it's separating the strands. So we have a very simple question. How does the DNA get inside? So the DNA gets inside with the help of another protein. Now, this ring is made up of 6 identical proteins. So let me show you that protein. So it's a protein called DNAB, and it has 471 amino acids. And we think of this, and its amino acid sequence is laid out here, and some of the functions are listed. It basically has 2 parts, one in pink, one in blue, and then these two parts are linked together with a green piece here. So this is what we'd like to understand in 3 dimensions. And through experimental techniques, we learned that this is what that looks like. So this is in 3 dimensions. The pink thing here is the thing that looks like a mushroom over here. This is the amino terminus. This right here is the blue thing. Looks like a sphere. The mushroom and a sphere. And then there's this thing that looks like a helix. That's just the green thing that links. So this is one copy of this machine. There are 6. And there you see the 6. The spheres pack into a layer, and the mushrooms pack into a layer. And this act of putting them all together makes a chamber inside, and this is where the DNA is connected. So this is the closed form of this protein. And now we'd like to understand, how does this protein get open? So when we started our work, we had the closed form. Another group determined the structure of the closed form. Here it is in cartoon form, the spheres and the mushrooms, central chamber. And then the same group had determined experimental structure, very challenging determination. And now you can see that there's a bit of DNA in the middle. Okay, and then this is still closed, the chamber is still closed, so DNA is inside, it can't come in and out. And so our question is, how do you open the ring? How do you put the DNA in, and how do you close the ring? And so other groups had worked out that one protein is involved in this. And so we took that protein, and we want to now determine its structure. The two together, see the two together. So a doctoral student in my group, Jill Chase, she took this on as her doctoral work. And I won't get into what the meaning of these patterns here. I will just say this is very good news for Jill. She's finished her work and graduated, but at the time this was a sign to rejoice. So she took all these images and put them together into a 3-dimensional representation of the molecule, and that's what you see here. This mesh here is what we get out of electron microscopy, and it's a 3-dimensional mesh, and it has internal information about the molecule. Now, this was a blurry view. This is kind of like the first image that I showed you. You can kind of see the outlines, and we can see things inside, but it'd be very hard for us to interpret. So what we did is we took a structure that had been determined already previously, and we gave the 2 of these to a computer, and we said, put this over there for us. So the computer did as it was told. It positioned the spheres and the mushrooms, the 6 of them, and the green linkers were all represented. And then we saw that there was a whole bunch of gray mesh that didn't have anything in it. And this is the second molecule that's missing. Now the second molecule, we didn't know what it looked like. And so Jill had to build this by hand. And so what we got was the helicase here plus its loader, the thing that opens it. Now what you can tell from this is that what used to be a ring is now not a ring, but it's been cracked open as a spiral. You can see there's an opening here. And you can see that a little better here. So this is the way we represent the structure. The blue and the pink are the sphere and the mushroom domains, and here's the opening, and then the loader is sitting here. And if we here you can see it spins around. So this is one of the layers. The opening is right here, and that's the mushroom layer, and the sphere layer comes in here. Here you can clearly see that it's open, and DNA can now come in and out here, and then the 5 loader molecules. And if you're a fan of science fiction, this may remind you of something. The loader sort of sitting on top of the helicase. When I first saw this, I thought of the movie Alien, and they call this the ovomorph. So this is before it becomes the thing that's going around. This is right when it's born, and it needs a human host to be born. Now, where does AlphaFold come in? So AlphaFold is a computer program, and it was developed by something called DeepMind, which is part of Alphabet, Google, and it can make predictions of protein structure. Now recall that there are about 200,000 protein structures in the database. So that's the data that AlphaFold was given, plus some other information. And through the AI process, it's now able to make predictions, and there were 2 publications in 2018. In 2021, they produced models for every protein that was known to science. So how do they do it? It's obviously we can't get into it today, but what this program does is it takes the input amino acid sequence, here it is here, and it uses a bunch of these, some of them from different organisms, chicken, rabbit, fish. And so if a protein is related in other organisms, it can use that information together. And there's a lot of things going on in the middle. And then at the other end comes the 3-dimensional structure. You get the 3-dimensional structure, and then it's colored by how accurately the program thinks it got the structure. So this is now revolutionizing our work, because for the first time in history, and you're the first generation to just go to a computer and type in amino acid sequence and get a structure, we can now do this. Now, how does this help us? Now, so it helps us because we did this structure in 2018, and so what you have here is the helicase, here are the loaders, the little aliens that have jumped on, and then we don't have the DNA in, but the cylinder here represents where the DNA is. Now, there's enough space for the DNA to come in and out, and this is a model we proposed in 2018. Now, what happened there is our maps were not so clear, particularly in a region here, that we didn't interpret. Now, with slightly better maps, we could now see that there was something in here that we missed in 2018. We couldn't, it was still blurry. We couldn't interpret it. And let's see, let's get these guys to think about. So the thing in orange we missed in 2018. With a slightly better camera, so to speak, we could see that this is now here, but we couldn't interpret it. We couldn't build a model. So this is where we took the AlphaFold model, calculated it. Now we know what it's supposed to look like, and then we pattern matched. We just matched the calculated structure into the density. It fit nicely, although we had to make adjustments. And then we could model the rest of it. Now the insight here is that although the ring is opened, and there's enough room for DNA to come in and out, the loader actually has closed off the path. This is a new insight that we obtained that we couldn't see 5 years ago. So there must be some additional change to get the loader, this little gate here, to come out of the way and to allow DNA to enter. So I'm past my time here, so let me finish up by just showing you the folks who did the work. Jill started us, and Dante Brown and Adipsha Shakaruba are current doctoral students with me who are continuing the work, and these are members of my group, past and present. I should thank everybody, particularly the people who supported us financially. And then I want to end by just telling you about some programs that we have at City College. One is a summer research experience, and we're about to start that program this summer. We've admitted our 10 fellows, and the fellows are embedded in research labs for the summer, and they work with us for the summer. And this has been running for 7 years, and most of the graduates go on to work in biotech. They go to graduate school or medical school. And then something new that we're doing this year, next fall, is starting a post-bac program where we focus on recent college graduates who may not know what they're doing, may not have the lab experience. And this came out of COVID when no one could work in labs.